Before we move on to the next part of the video, I should tell you one more thing. So between this synapse over here, the terminal bouton, and this is the membrane of the muscle, there is a basement membrane that connects them over here. Now in this basement membrane, basilar membrane, there is an enzyme called acetylcholine esterase enzyme. So what it does is it breaks down your acetylcholine molecule. So when you release acetylcholine molecule, acetylcholine esterase will destroy this. There's another enzyme called pseudocholine esterase, but we'll talk about that later in the clinical aspects. I want you to focus on this acetylcholine esterase. It breaks down acetylcholine by hydrolyzing it through a hydrolysis reaction. That's the meaning of esterase. It takes a little molecule, so there's an organic molecule, whatever it is, and what it does is it will combine one water molecule, break it over there, so H2O, so there is OH. OH goes one side, H goes to the other side, molecule is broken, and so is this molecule. So that is how you make esterase. So that's an important enzyme that we will be talking about in the chapter on myasthenia gravis and Nathan-Lambert syndrome, coming up now in the next video. Through the events taking place at the neuromuscular junction during transmission. So here we have the alpha motor neuron. Step 1, action potential travels down the alpha motor neuron via saltatory conduction through the nodes of Ranvier towards the terminal bouton. We are only looking at one of these terminal boutons. Number 2, calcium enters into the cell. Why? Because the sodium enters into the terminal bouton, depolarizing the membrane of the terminal bouton, causing the opening of voltage-gated calcium channels, which are present over there. Then after that, the calcium entry in step 3 will cause vesicles, a cohort of vesicles up to 60 to 120 to move towards and dock with the presynaptic membrane. So a bunch of these vesicles that are selected, a few of them, like only 60 to 120, will fuse with the cell membrane. They will be selected, they will start moving towards the cell membrane. And don't worry, we have 100,000 other vesicles which are still over there in each terminal bouton. Then after that, step number 4, after the docking takes place, so the vesicles move and they fuse with these red regions I told you, the T snares and the V snares. Watch the previous part about the structure of the neuromuscular junction. Anyway, so it fuses, the V snares and the T snares fuse, docking takes place over there. And then step 5, exocytosis. Acetylcholine is released. Acetylcholine is released into the synaptic cleft, okay, from the vesicles due to exocytosis that has taken place. And this is going to now, if you go across the synaptic cleft over the distance of between 1 to 100 nanometers and quickly fuse with the receptors on the postsynaptic membrane. So if we look at the receptors in postsynaptic membrane, it's this these green ion channels, and they are the N1 or NM nicotinic cholinergic acetylcholine receptors, ligand-gated ion channels over here. So sodium enters into the cell, and this area where you see the crests and troughs, this is the postsynaptic membrane. Sodium starts to enter into the cell from the ECF into the ICF, and it makes the cell positive. You are creating what we call an end plate potential over here. So we are getting something called an end plate potential over this motor end plate, this area which has got the crests and the troughs with the voltage gated, with the ligand gated sodium channels. And then what happens is, if you have released enough acetylcholine and they find their way to enough number of channels, then it will cause enough depolarization to take place. If the depolarization happens from minus 90 millivolts RMP towards minus 65, meaning you made the cell a little more positive by up to 25 millivolts, then you can initiate an action potential which will spread to the surrounding areas and spread along the sarcolemma down to the T-tubules and cause the excitation contraction coupling, which we will deal with in the next topic. Then what happens is the acetylcholine must be destroyed as soon as possible, and this is because if you leave acetylcholine on these receptors, the N1 receptors or the NM receptors, they will actually downregulate the receptors. They will not respond so well. They actually get desensitized in a way. So you don't want this to even last for a few milliseconds more than is necessary. So once the acetylcholine binds to the receptor, it is immediately destroyed by acetylcholinesterase enzyme, and that terminates the signal. To the fusion of these vesicles, we should also just talk a little bit about it. So here is a vesicle that I've shown over here with acetylcholine. You can see I'm underlining it over here. So 10,000 acetylcholine molecules are in one quanta, and these acetylcholine molecules are surrounded by these receptors that I'm drawing like V-shaped receptors. These are your V snare, V for vesicles, because they're coating the vesicles. They mostly have two names, synaptobrevin and synaptogrammin. These are the main ones you should know about. There are many other molecules involved in this, but we'll focus on the main ones, synaptobrevin and synaptogrammin. They belong to the V snare category of proteins. And there's another protein called RAB3. It's a kind of G protein receptor that senses the calcium, and that's all what you need to know about it. So the vesicles under the influence of calcium will move towards the active zones, and these are the zones which have got something called T snare over there. T snare is the target snare protein. When the V snare and the T snare fuse, because they were pushed over there by the calcium, then then you get exocytosis, and T snare is divided into syntaxin and SNAP25. These are the main molecules over there. Okay. The clinical aspects of neuromuscular transmission. So there are various disease conditions we'll be speaking about. The first one I'd like to talk about is myasthenia gravis. So myasthenia gravis is an autoimmune condition. So that means the immune system starts attacking some component of the very own body it belongs to. It's not a good thing, because destruction of useful parts of the body might cause some damage and loss of function, and eventually lead to disease and suffering. So now we have autoimmune condition, myasthenia gravis, and in this case what happens is you make these antibodies that float around, and they are particularly going to destroy something called the NM receptors. Remember your NM receptors, the nicotinic acetylcholinergic receptors over there in the postsynaptic membrane? Yeah, the ones which I've drawn in red over here. These on the postsynaptic membrane over here are the NM receptors. So these antibodies would like to go, these myasthenia gravis antibodies, and they like to destroy these receptors. So what happens is you eventually run out of receptors in the postsynaptic membrane. Sorry about that. It just was a silly thing. Anyway, so the point is you are going to run out of the total number of receptors for your acetylcholine. So when the neurotransmitter is released, acetylcholine, after an action potential is formed and it goes down the membrane, and you know the sequence of events. Sodium comes in the nodes of Ranvier, and over here we have voltage-gated calcium channels. Calcium enters into the cell. A few of these vesicles, like around 60 to 120, will exocytose their acetylcholine quanta. Now, you must understand that you need only, I said we release 60 quanta, or 60 of these vesicles. Each vesicle has got around 10,000 acetylcholine molecules, so you can do the math about how many molecules are released from each of these terminal arborizations in each synapse. But what I want you to know is that you can actually elicit the action potential in the postsynaptic membrane if you just release 1/10 the number of the quanta. Just 6 quanta are enough. That means our body released 10 times the number required. So you are guaranteed to get an action potential in the postsynaptic membrane. This is called safety factor. Safety factor. Now what happens is we have a safety factor of 10 times because we are releasing 10 times the number of vesicles that are actually required to release action, to cause an action potential. But when you make this auto antibody which will destroy your receptors, now the acetylcholine that gets released, it cannot find these nicotinic receptors, acetylcholine receptors so easily. So it takes time for it to find the molecules. The ligand has to take time to bind to the receptor over here. It will take time to find this place, and there won't be enough number of receptors. And so while it is wasting its time finding these receptors, the enzyme called acetylcholinesterase is going to, which is present in the synapse, is going to destroy acetylcholine. So it's going to break it into the acetyl and the choline component, and the choline will get it will get taken up again into the neuron and used again. So now what happens is this acetylcholine is going to get destroyed by the acetylcholinesterase, and since there is no movement taking place, the brain will still give a bunch of action potentials down, and you will start releasing more and more of these vesicles contents, more and more quanta, until you exhaust the vesicles. You see, you have around 100,000 vesicles on average in each of these terminal boutons. Now if you keep giving a chain of action potentials, you're releasing way too many quanta, more than you are able to replace per unit time or refill per unit time. So what's going to happen? You are running out of neurotransmitter, you are running out of neurotransmitter and the vesicles that store it, because these receptors for the acetylcholine are destroyed by the antibodies. Now that's going to cause weakness and paralysis. So what happens is the person who comes with myasthenia gravis, he is going to be fine in the beginning, but if you make him do a repetitive movement, especially a repetitive movement, something like, for example, combing the hair or chewing food. So chewing food, what you call mastication, or combing hair, to run a comb through the hair, you're doing the movement multiple times again and again. So what happens is, to make one movement take place, you release a lot of acetylcholine vesicles, and the movement won't be so easy and so strong. And as you keep repeating the movement, you will deplete your vesicles, causing the paralysis and weakness of the muscle. And one more very important thing that happens here is ptosis. Ptosis is the drooping of your eyelids. Your eyelids will droop. Like if you remember in your childhood, there was a cartoon with Tom and Jerry kids and Droopy the dog. So if you remember Droopy the dog, he used to always have these drooping eyelids over there, and Droopy and Dripple. I don't know if you know that. But the point is, your eyelids are going to be, they're going to droop. Ptosis. Repeated movements cause fatigue. Okay, fatigue, and this will be similar to what you call lower motor neuron type of fatigue, paralysis, which it looks like paralysis. Eventually you will get paralysis, and this is going to look like lower motor neuron type of paralysis, and we'll talk about this in CNS. I just want you to know that the muscle becomes totally loose, lax and flaccid. So that is your paralysis that takes place over here in myasthenia gravis. So this is usually associated with a condition called thymoma, meaning a cancer of your thymus gland. So your thymus makes a lot of the T cells that are like the boss. They tell the other immune cells what they're supposed to do. Now if you have a cancer or a tumor in your thymus gland, then you're not going to be making the correct kind of T cells. It won't function properly. So there's a chance that some bad cells are produced in the thymus, which will make these antibodies that will destroy the nicotinic receptors over here, reducing the safety factor and causing all these problems where we are exhausting the acetylcholine. There is a reduction in the available vesicles eventually. So your repeated movements become weaker and weaker, and that's very important. Even when they talk, even when they speak, They speak in the beginning, when they start talking after a nice period of rest, they speak strong, then eventually it becomes weak. Sometimes the paralysis is so bad, even the respiratory muscles can get affected. So they can get respiratory paralysis and they can die from this. There is a chance of this happening over here. And if you cut the thymus gland, then most of these people just remove the thymus gland, thymectomy. Up to 85% of these people will get cured without the need for treatment. Otherwise, what you have to do is you have to give certain drugs. So the drugs, this is the symbol for drugs, RX, it's that it's an invocation to one Greek god, I think it's Juno, and it means take thine medicine. So that's what all these pharmacists write. So this is the medicine we give. And what are these? These are a class of drugs called acetylcholine esterase inhibitors. How does this work? Well, remember acetylcholine esterase over here? This enzyme breaks the acetylcholine. So you're not giving it enough time to find the few receptors that are present. So what happens is, if we give this enzyme inhibitor, if we remove this enzyme from the equation, now what's going to happen is the acetylcholine is going to be able to find these receptors over here and eventually make an action potential take place. At least that's the hope. And it does happen. So we have drugs like physostigmine, or neostigmine. They play a role. Also, immunosuppressant therapy with steroids, corticosteroids can also be given. So that suppresses the immune system, which makes these antibodies, which are the villain over here. So that can also be used over there. We can use corticosteroids when things get really bad. And also we can sometimes do a procedure called exchange plasmapheresis, where we take your blood, separate the blood cells from the plasma, and now the plasma is going to contain these antibodies. So we'll just get rid of that plasma and replace it with some healthy substitutes instead. So that's how it works over here. This is your myasthenia gravis. The other condition I want to talk to you is something called Eaton-Lambert syndrome. Now here we have again another autoimmune condition, but how does this autoimmune condition look now? Again, you are making antibodies over here, but to which component? It's not going to be the nicotinic receptors over here that get affected. It's not the NM that gets affected. What gets affected instead is the voltage-gated calcium channels. So it's the voltage-gated calcium channels that are attacked over here, which I'm drawing in red for your benefit here. So these get attacked by antibodies. And this condition is associated not with the thymoma as much, but rather with another condition called small cell carcinoma of lung. Small cell carcinoma of lung. So you get this lung cancer, and the lung cancer actually does something else. It doesn't just grow and damage the lung and eat up all the nutrients which should go to the healthy tissues. Tumors can do other things. Sometimes they can release hormones and chemicals which are not required because it's unregulated. So these tumors can sometimes make antibodies. In this case, we induce the production of these antibodies that are going to damage your voltage-gated calcium channel. So if you don't have enough calcium, remember, calcium comes in, something goes out. You don't have enough calcium coming in the cell, you don't release enough acetylcholine. You cannot release acetylcholine for that reason. It won't be enough, at least. So again, you are causing a reduction in the safety factor. Safety factor becomes less over here. Now, when you look at these people, they will start with fatigue, and they will be fatigued, like their muscles are almost paralyzed. It doesn't move so easily. The movements are weakness. But as you tell them to start working their body and telling them to move around, they start with one movement, and some of the calcium channels that exist that have not been damaged will cause some calcium influx, some. So the first movement might be a little weak, but then you give another action potential, you do the movement again. Now there's already some calcium that has come inside from the previous action potential. Now what's going to happen? You're going to give another action potential, and again more calcium comes in. So it's adding up. Calcium is sort of summating. It's adding up in the, it's accumulating in the presynaptic area over here, in the terminal boutons. So you start weak, and then the next movement becomes a little stronger and strongest. So your movements are weak in the beginning, then they get strong later. Even when they talk, when they speak, if you want to differentiate between myasthenia gravis and Eaton-Lambert syndrome, the Eaton-Lambert syndrome patient is going to start very weak. He's going to talk very faint. His voice is going to be very faint. So his voice is going to be very faint. Then later as he talks, it's going to get louder, like, doctor, I don't know, I feel so weak. But then eventually my voice gets good like this. So, so the voice goes from faint to strong or loud. So from soft to loud sounds. In myasthenia gravis, sometimes the person is speaking, and later he gets weaker and weaker and weaker. So the muscles, like the breathing, respiratory muscles, the diaphragm get weaker and weaker because of repeated movements when you're talking. And all these movements for talking also get affected. So again, you can get paralysis over here. And here we have, again, certain treatments. Like we use those similar medicines we used before. From immunosuppression, like the corticosteroids, and even the use of acetylcholinesterase inhibitors. They can also be used over here. There is also another class of drugs called aminopyridines. But first remember, we use these acetylcholinesterase, it's just not for the treatment, acetylcholinesterase inhibitors, like neostigmine, physostigmine. You can use corticosteroids. There are other immunosuppressants out there in the market, but these are the main ones you need to know about. And also there's a class of drugs called aminopyridines that are able to increase the amount of calcium that enter into the presynaptic membranes. So they cause more accumulation of calcium. So this calcium accumulation helps in the release of more quanta. So you're sort of trying to release enough number of vesicles and overcome any paralysis and weakness that exists over here. So that's your Eaton-Lambert syndrome. Then there's another condition called botulinum toxin poisoning. Botulinum toxin. And there's poisoning and there's clinical use of this. So there are two ways you can look at botulinum toxin, where you on purpose inject it into your body to cause muscle paralysis, or where you would unknowingly consume some packed up, usually airtight canned food over there from the market, and eventually damage your health. So we'll be talking about that.